So, hello everyone. Welcome to another session of RSGB Türkiye. So, it is one of our monthly seminars, and beginning with our social media accounts, there's slides shows you our several social media accounts. We welcome new people to our group, and we would like to further enhance and enhance our community and work with new people. So today's presenter is Assistant Professor Francesco Gentile, and he's from University of Ottawa in Canada. And the topic of the presentation is diving into deep chemical space to advance ligand discovery. So stage is yours, Professor.
All right, thank you very much, Dennis. Let me just share my screen. And can you see?
Yes, we can see.
All good? Perfect.
Yes.
Okay. Okay, thanks very much for this kind invitation. I'm very honored and pleased to be able today to present a bit of the research we do in our group. So my name is Francesco. I'm an assistant professor in chemistry and biomolecular science at the University of Ottawa and a principal investigator at the Ottawa Institute of System Biology in Ottawa, Canada. Before I start, I'd just like to thank a bit the wonderful people in our group. They do most, if not all, the job that I'm going to present today. So the merit goes to them. As you can see, it's a pretty it's a growing group, it's a diverse group with researchers coming from literally any corner of the world and spanning.
Dear Professor, we cannot see your like the connection was problematic, so we couldn't
You
hear
can
you. At last, you say that there is various people from different countries.
Okay, can you hear me now?
Yes, I can. We can.
Okay, so it's you can see and you can hear?
Yes, we can.
Okay. So our lab is located in the STEM building, University of Ottawa. You can see in the picture which face the Rideau Canal, was one of the UNESCO World Heritage Sites of Canada and the longest ice rink for skating in the world in the winter when it freezes. Now, after this little information about the lab, I'd like to introduce a bit the talk of today. So since since I would like to talk a bit about exploring the chemical space, start with a fast introduction about what the drug-like chemical space is and what's its use in drug discovery. So to put it simply, in the early stage of a drug discovery campaigns, when you have a drug target in mind and you would like to identify chemical matters that can react with that target and induce some desired biological response, it is very important to identify what this chemical matter is, so to identify what is called a hit compound. So hit discovery is one of the critical steps in early stage drug discovery because those compounds that are identified as active are then what hopefully one day will become your drug candidate. And one way to identify compounds that are reactive against a certain target is to screen physically a chemical library. So take a chemical library of synthesized molecules and screen that exhaustively against cells or protein, depending on the assay you want to use. Of course, due to the limitations arising from the cost and from the scalability of high throughput wet lab screen, we can state that it's hard to go for more than few millions compounds, even for big pharma and national laboratories. This is paradigm that is changing with recent introduction of DNA encoded libraries that perhaps some of you may have heard of. although they still present some technical limitation that prevent to exploit their full potential. So I think this will improve and will enable to experimentally test chemical space of billions of compounds, but at the moment, other alternatives are highly sought for the exploration of the chemical space. So since the early 2000, there was a development of the so-called virtual libraries, which are repositories of repositories of commercially available molecules that can be interrogated with computational methods, and then they can and therefore the molecules can then be purchased and tested. So this was pioneered by Zinc, and Zinc started with 700,000 commercial compounds, and right now exceed 40 billion compound in its fully enumerated version. And this is because around 2016 there was a so-called explosion of make-on-demand libraries of synthetically tractable molecules that are not in stock for chemical vendors, but they can be synthesized rapidly for a limited cost. So some of those libraries, you see the figure here is 10 billions, but actually some of them are already exceeding the trillions of molecules because they grow with the number of fragments that are available and the reaction that can be used to combine those fragments. Even so, we are very far from exploring completely what is the theoretical drug-like chemical space, which was estimated to be 10 to the power of 60 drug-like molecules, 10 to the power of 24 that could be synthetically accessible by known methods in 2022. So I'm sure that is now. So we are still far from exploring the entire chemical space with commercial chemical libraries. In parallel with the emergence of these virtual libraries, computational methods have been developed to triage those libraries. And nowadays they play a central role in modern drug discovery pipelines, being them AI-based, such as AlphaFold methods, or physics-based, as like conventional docking. The idea is to start with a drug target and then in virtual screening, screen a chemical library, a commercial chemical library, compounds that can be obtained easily using some kind of computational method to perform this high-throughput virtual screening. And the goal is to identify a narrow subset of molecules that are promising from a computational point of view. They can be tested to identify it, that they can then be optimized with MedChem techniques and using computer-aided drug discovery methods to support that until you hopefully reach a preclinical drug candidate. Now, high-throughput virtual screening based on docking is one of the most used methods, especially when you don't want to base your screening on previous known ligands for a specific target. And docking relies on fit small molecules in an energetically favorable way within a rigid or semi-rigid binding pocket. And it can take between alpha second to 120, so 2 minutes, depending on the number of rotatable bonds, the ligand, and the algorithm that you are using. This is, I think, this time estimate still also with the new AI-based docking methods that have been proposed recently. Now, when docking was first applied to these ultra-large libraries, so the libraries that start to emerge after 2016, he had great promises. The first papers that were coming out around 2019, 2020 demonstrated that docking could provide a clear advantage compared with the docking ultra-large libraries can provide a clear advantage compared with the small in-stock libraries that were screened until that point, as it can identify compounds that were more potent, more diverse, and novel compared with what you could find inside in-stock in-stock catalogs. And moreover, this large library also allow you to do rapid expansion, analog by SAR expansion of the hits that you will eventually find. But of course, the computational cost to screen those libraries is prohibitive and keep increasing with the size of the library. If we were to screen a Zinc 2020 that has 1.4 billion molecules using Glide, a very popular docking program using 640 core GPUs, it will take 300 days just to screen a library computationally, which added to the uncertainty that you always have when using a computational method to identify molecules, is certainly not feasible. So the solution is to find alternative ways to identify the most promising molecules within these libraries, and or to have more computational power. So I'm going to talk about AI-accelerated virtual screening, but before that, I would like just to spend a few words on why physics-based virtual screening is a good oracle to train molecules on, because physics-based virtual screening based on conventional docking is not a train on any data, and is based rather on physics-based equation to compute binding affinity and geometries of the pose. And therefore, on first approximation, is suitable to identify new ligands for new targets, because it doesn't require any prior knowledge of anything that can bind to that specific target, as long as the pocket is tractable with the equation the underlying physics-based equation, and the ligands have been parameterized for that specific scoring function. So it's very suitable to screen these large libraries, as was proved by many studies. However, as I already mentioned, dealing with the ever-expanding chemical space is prohibitive, and therefore alternative methods to brute force docking be proposed. Two of the most promising one have been using fragment-based methods, where those libraries are decomposed in their original building blocks that are docked. Obviously, the original building blocks is order of magnitudes less than the fully enumerated, fully built compounds that you can obtain by combining the building blocks with the set of reaction that the vendor has. And then the compounds, the fragments that score the best, they can be then expanded with whatever is compatible within the library, or machine learning surrogates. Basically learning docking scores from a subset of molecules, and then predict the docking score of the entire library, and then discard whatever is the low scoring and keep just the top scoring molecules. I'm going to talk about the third strategy you can see here today. And I would like just to introduce a method that I contributed to develop while I was in my postdoc at the University of British Columbia, called deep docking. This is a method that relies on initial random sample of ultra-large library, the docking of these small sample of small molecule, and the training of a model that can predict the docking score class of the remainder of the library. And this process is performed iteratively using active learning in a way that at every iteration, a new set of molecule is proposed by the model's acquisition function to be acquired and labeled, docked, and then added to the training set, and the model is retrained or continue to train in order to improve the prediction. And this is because, obviously, if you start from just a small sample, you won't obtain like a reliable model right away. And this is pretty efficient in order to rapidly focus on the top scoring molecules within the library and identify them with a certain accuracy, molecules that are they are top scoring, or that fall within like a certain range of top scoring within the entire library without having to dock the entire library. We estimated that for billion size library, 1% is usually good amount of molecules that you can dock to predict accurately the top scoring one. And we also observed that the model was with certain discrepancy between a target and another, but was in general behaving pretty consistently in terms of reduction of the database across all the iteration of active learning, as the model discards more molecules as you become more confident and focus on the more top scoring molecules within the library. So we demonstrated it was working against different protein drug target families in a consistent way. Okay, so now this is interesting and is always good to optimize for docking scores. It's good to use generative models, which is another approach to navigate the chemical space to optimize by docking scores. But does this work? Like, does it make sense to screen large libraries against drug targets? And one of the main interests we have is actually to see if there is advantages in unlocking new chemistry and biologically active chemistry for targets that are either underexplored or deemed undruggable. In other words, if we can leverage the AI acceleration to screen larger libraries against those targets and identify compounds that give the biological response that we seek for. And ideally, we wanted to test these, and this is what I'm going to present today, mainly against the cold target that don't have any prior ligands, in order to properly assess how much physics space, the power of physics space in identifying new ligands when the scenario offers just that, because you can train on anything else. So one of our such project, one of the possibility of testing, stress testing this, was through a collaboration with the lab of Michele Vendruscola at the University of Cambridge, where we sought to identify inhibitors for the aggregation of the amyloid beta 42 peptides, whose aggregation into fibrils is one of the central features of Alzheimer's disease. And in particular, in the secondary nucleation, which is the mechanism through which a fibril that is already formed catalyze more polymerization of, on its surface, more polymerization of the amyloid beta peptides, is a particularly interesting mechanism to inhibit, because in order to treat Alzheimer, as the recently FDA approved or investigated antibodies for Alzheimer rely on blocking this kind of interaction. And since antibodies present some other challenges in terms of safety, looking for small molecule alternatives that could block selectively the same mechanism would be very interesting for developing new therapies against Alzheimer. Interestingly, the screening against the putative catalytic site for secondary nucleation, the virtual screening of in-stock libraries didn't yield any hits. So in this case, we were really looking into a scenario where the target is still considered undruggable, at least within stock libraries. We want to see if, like, by expanding to a much larger library, we could unlock chemistry that could inhibit successfully the secondary nucleation. So we focus on a molecular weight filtered version of the Zinc-20 database. This was to augment the chance to identify molecules that could be blood-brain penetrant. And we tweaked the original deep docking pipeline by replacing the steps that were performed using proprietary software, so licensed software, with open source alternatives, such as downloading 3D conformations directly from Zinc, and docking with open source Vina GPU. And this was made by a student in the Vendruscola lab, Michela Brezinova, that did much of this wonderful work to develop this open source platform. And we then applied to the catalytic site of the secondary nucleation on the amyloid beta fibrils. Through the iteration of active learning, we observed that the model was able to improve its classification performance, as expected by the active learning framework we were based on. This is in terms of classification of top scoring molecules within database. And we also observed that we were able to go pretty fast within 5 iterations by docking less than 1% of the library. We were able to go rapidly from more than 500 million molecules into few tens of millions that could be then docked in order to identify the true positive within the last set. So successful from the point of view computational, and somehow what expected even with this open source framework. Now the biological testing. So to our surprise, like pleasant surprise, more than 50% of the 39 compounds that were bought were actually active in slowing down the aggregation of amyloid beta fibrils, amyloid beta peptides. And the two most promising compounds, the one that have the longest normalized half-time, were actually subjected to more deeper investigation in order to confirm the mechanism of action via secondary nucleation, as well as to also check if they were actually binding to the fibril. And those compounds were not only inhibiting selectively secondary nucleation versus primary nucleation, but they were also tight binders to the amyloid beta fibrils with dissociation constant, the low nanomolar. And this was pretty surprising and and very encouraging, given that no round of medicinal chemistry optimization or analog expansion was performed yet. Those were compounds that were coming out from the raw screening using AI acceleration. Last but not least, the compounds were also able to inhibit aggregation, secondary nucleation-induced aggregation of amyloid fibrils in cell culture, in neurocellular cell cultures. So showing the biological activity also in a more complex system such as cell model. Another project that we have tried to apply the same paradigm via accelerated screen to target a cold challenging target was the investigation of allosteric inhibition of the erythromycin ribosomal methyltransferase C enzyme. This is RMC is a bacterial enzyme that plays a key role in erythromycin resistance by methylating the ribosome and thus pushing erythromycin out from the binding site and being responsible for acquired resistance to erythromycin. In this light, targeting allosteric sites rather than the canonical SAM binding sites is very attractive because this would allow to first not compete with the high cellular levels of SAM within human cells, and also would also allow to develop compounds, scaffold that are more selective than SAM binding compounds, because the SAM binding site is conserved also around across human methyltransferase rather than in addition to the bacterial one. So collaborating with the lab of Ruchi Anand at the Indian Institute of Technology, Bombay, we sought to screen 83 million from the enamine diversity set, which is like a diverse, smaller representation of the entire enamine real database, the ultra-large library, most famous ultra-large library of enamine, using accelerated vitro screening against a allosteric region that was identified by the Anand lab via structural biology. And then the 60 compounds divided into cells that were binding to two different regions, two different subpockets of this allosteric region were purchased and tested in a methylation in vitro assay. And here, more than 50% of the molecules turn out to be methylation inhibitors at a concentration of 10 micromolar, and 6 out of 60 were potent methylation inhibitor, being able to inhibit methylation in the in vitro assay at a for more than 50% when a concentration of the compound was 1 micromolar. So those compounds were then tested for dose response curve, and the most interesting one, the most interesting two chemotypes were binding at each to one of the two different subpockets of the allosteric site. The most interesting one on the pocket that we name pocket 1, display like a mid nanomolar IC50 in terms of methylation inhibition, and the one binding on pocket number 2 was showing a low micromolar inhibition. And these were compounds that were coming straight from the screening of a diverse library, no analog expansion or no Medichem optimization. Also, again, we sought to test if like those compounds were also like of biological relevance, and we saw that all the compounds that were showing inhibition or methylation more than 50% at the methylation assay at 1 micromolar were actually able to sensitize Staphylococcus aureus to erythromycin significantly, with the two compounds that I introduced here, C9 and C23, being the most noticeable one. So again, like a good validation of AI ultra-large screening against targets that have no prior of known ligands. Another exercise we took to stress test like this kind of methods participate to the cache challenge. So I'm sure some of you are familiar with that. Those are a series of open science drug discovery challenge where a committee made by industry and academic members select a number of challenging drug targets, and those are proposed to the participant in order to identify commercial biochemical compounds using their own peer-reviewed methods. And The goal is to identify both the baseline and the state-of-the-art init identification by then testing those compounds, identify the one that can identify the workflows that can return the best compounds. The first challenge was against the WD40 repeat domain of the leucine-rich repeat-rich kinase 2, which is the most mutated gene in Parkinson's disease, and the WD40 repeat domain is an emerging pharmacological target within this large protein. And the goal was to identify molecules that would bind to the central cavity of this domain. And at the time, I was still in my postdoc, toward the end of my postdoc, so we collaborated with Oleg Saizayev at the university, at the Carnegie Mellon University, and Maria Kornikova in the same university, and with my postdoc advisor, Al Cherkasov at the University of British Columbia. And here we combine deep docking, so the screening of the compound using accelerated docking with different selection methods based on consensus docking and expert selection. So like we want to see if we were able to identify better compounds by using an expert interrogating the poses coming out from deep docking, or by using pure automated consensus between two docking problems. And we combine also these with molecular dynamic simulation, especially absolutely binary free energy calculation at the end to triage the last set of molecules that were then submitted for purchasing for experimental validation. And from molecular dynamics refinement of the potential leads, the first observable we saw was that most of the compounds were stable within the binding pose within the binding site in the pose that was predicted by docking, although some of them were displaced significantly. They were showing an RMSD of more than 3 Armstrong between the pose that was obtained at the end of molecular dynamics simulation and the one that was predicted by docking, so suggesting that those poses were rather artifactual and not realistic. This could be like a excellent way to filter docking results, and perhaps something that we should try to predict in addition to docking scores. Not really surprisingly, there was no correlation between the delta G, the absolute binding affinity delta G, and the standardized, the consensus docking score that we obtain, and this was not really expected, as especially the docking scores. It would be unrealistic to expect that they can really correlate with real binding affinities, and even absolutely binding free energy is debatable how well they correlate for targets of this level of difficulty. And we usually sought for molecules that are at the intersection between two scoring methods rather than like a really correlation between the two. So like whatever works well in both methods is usually more promising. This is also the basis of consensus in general. Okay, so the compounds were purchased and tested with rigorous biophysical binding assays by the organizer of the CASH Challenge at the University of Toronto. So out of 59 compounds that were obtained, 5 of them were working. So like 8.5 hit rate, 8.5% hit rate, respectable for a target of this type. And when we expanded those molecules, 9 out of the 32 analogs turned out to be to show a dose response in a sulfur plasma resonance experiment in the second round, therefore confirming that there was new chemical series that was identified, and this was one of the first chemical series that was confirmed and discovered for this target being a completely cold target. Since this was part of a challenge, our workflow was also deemed to be the top ranked one. It was voted at the top rank by the experts that were evaluating the compounds that were discovered, the top ranked one, so formally the winner of the challenge. Having tied the first place with another team from the University of Pittsburgh. Then we decided also to participate in the CASH tree challenge. This time it was a different scenario. The goal was to target the SARS-CoV-2 non-structural protein 3 macrodomain, that has been investigated before. There were like a series of publications reporting both fragments, lead and full compounds that were binding there. Crystallography was performed on many of them, in a crystal structure is known. However, the best compound were identified before bear a carboxylic acid moiety, and therefore negative charge made to interact with one of the hotspot within the binding pocket. And this was not desirable because it could yield some problem with cell penetration. So the goal of the challenge was not only identify new compounds and binders for the macro domain, but it was also to identify compounds that don't bear a charge moiety. Again, here I was in the transition between my postdoc and my assistant professorship, and I team up with our group. We team up with Art Cherkas of the University of British Columbia to perform the screening against this. We use a consensus screening pipeline here, a bit different than before, because we want to explore more diversity at the beginning rather than go right away into ultra large libraries. So we focus again on the enamine or the real diversity set. This was 83 million molecules, as I already mentioned before. We then remove molecules with more than 2 chiral centers. This is just a quality control that we always do because chasing highly chiral molecules that are provided as a mixture obviously will dilute the potency out a lot. And then we use glide to screen the molecules. And selection of the molecules that turn out to be more potentially more promising on the eye of a medicinal chemist. And then we have a combined set where we have the expert shortlist and at least 2 votes in our consensus scoring workflow. And these are either 10 molecules. And then we propose the hits to be tested. So this time the results were very different than the other campaign I presented earlier. Just 1 molecule turned out to be a hit, and this was also this very low hit rate was across all the participants in the cache challenge, which is surprising despite because the large amount of structural data that was available for this target. And this was from the combined set between the consensus and expert selection. The good news was there was no carboxyl group or other charge groups. However, as you can see, the molecule was also not, there were like a central group that was resembling closely the one in the original crystallographic hit that we considered. And this is probably most likely because obviously there is a bias in the screen that was introduced by using the crystal structure of that the crystal structure of the complex with that compound. The organizers were also able to crystallize the hit that we identified, and that confirmed the predicted binding pose, which was a very excellent validation of our screening pipeline. Because crystallographic validation is obviously the ultimate validation of any structure-based method for discovering units. We then proposed 16 analogs following a similar screening protocol, but applying that from a much larger library to the enamine real. And all the 16 were displacing the substrate for the binding site we're targeting, and 12 of them were confirmed by SPR. The good news again is there were no charge groups and they were selected for MAC1 against other targets that were tested. The potency was not improved much, but these are under chemical series, non-charged chemical series that can be treated, it could be further developed. And again, even in this case, we were deemed to be the winner, the former winner of the challenge, as we secured the first place across all the 25 teams that participated there. Okay, so I'll use the last minutes that I have to talk a bit on something beyond the selection. So far I talk about using AI to accelerate the physics-based calculations, physics-based simulations, and how this can leverage the speed of AI and the generalizability of physics-based methods. But now I would like to talk a bit about another research topic that we are very much involved in exploring, which is how we can leverage, in this case, physics-based methods to generate ensembles where AI can learn more accurately protein ligand binding. So before introducing this idea of a pose ensemble graph neural network, Both physics-based and AI-based docking rely usually on generating multiple poses, multiple complexes that are independently score ranked. So to each pose, there is a binding affinity that is associated with that, or a binding probability associated with that. This means that 90 to 99% of the poses that are generated are usually discarded without further analysis. However, what we realize is that first, there is a significant gap between sampling and scoring. Usually docking algorithm, again, physics-based or AI, are very good at sampling poses. You can identify often the crystallograph, the low energy pose, the lowest energy pose, the crystallographic, the pose you will see with the crystallography within the ensemble that is generated by docking, but is not that often ranked as the first. Therefore, discarding all of them seems counterproductive. And the second observation we made is that there was a way to leverage the information of the incorrect poses, the one that are not the lowest energy, the one you wouldn't see in crystallography, but they are at least physically feasible because the algorithm predict those, generate those. And we could also think of them as like as an extreme case of out of equilibrium poses that are very rare to observe in nature, but perhaps can occur. So Tanawat, who is a postdoc in my laboratory, developed this concept of post-ensemble graph neural networks, where a graph neural network framework is used to capture conformational ensembles. We use docking to generate those. So each node in the graph neural network represents one of the poses, and they are connected by the RMSD difference between one pose and the other, used as a collective variable to connect two different states observed from docking. And then this model is used in a multitask prediction framework where at the node level, we seek to predict node probability, the likelihood for the pose to be realistic. So to be the below 2 Armstrong from ideally crystallographic one. And the graph level prediction, where all the poses are taken consideration after the readout of the graph, we use that to predict the PKD. And this is obviously to take advantage of the entire ensemble to predict the dissociation constants that are thermodynamic and therefore ensemble based properties. So a different way to compute to fit dissociation constant to conformation ensemble rather than single poses, as some of the docking program do. Interesting in terms of docking power, our method, the method that Ottawa developed, show significantly improved in performance compared with the different physics-based tools used both in rescoring or docking settings. And when we interrogated this method also on data leakage robust test sets, or test sets that were not seen by the new docking AI-based docking algorithm, we also observed that our method was outperforming or outpar with other deep learning methods that use significantly more complicated models and features, because our method utilizes just energetic features extracted from docking as a feature, so no coordinates, no complicated features. Moreover, we also check how well this model will be able to model dissociation constant that are experimentally known, although with uncertainty that is always associated with this kind of experimentally measured quantities that can vary a lot if measured in different laboratories. Nevertheless, we observed that the method was actually outperforming all the other scoring function that we use in terms of correlating with PKDs that were experimentally PKDs, although the correlation was just 0.38. This is, again, in my opinion, this is more of a problem of how noisy the experimental data may be than like a problem with the mods that can learn that. And anyway, dissociation constant, this turn out another confirmation dissociation constant are hard to learn in a regression setting. But the predictive index, which is a quantification of how well a model can rank active ligands with different power, was significantly higher in the case of our method that we named DogBox2, DBX2, compared with all the other methods. Now, you will see that the predictive index of KarmaDoc and CarsiDoc are pretty low and the R-square too, but those two deep learning methods were not trained or developed by any means to predict binding affinities. So it was just like a routine comparison, but is not something that speaks bad about those methods. They were not trained for that. They were trained to generate poses, not to score them. We also test that on the LeadPCBA dataset on proteins that were not in the training set, and we so in this dataset you have active and inactive molecules rather than active decoys, so a more realistic virtual screening or high throughput screening scenario. And we selected the 3 targets that were not in our training set, and we interrogated all the models that we have performed a comparison against in terms of docking, as well as like a DocBox 2. And we saw that our DocBox 2 was able to outperform both the graph level and the node level scores. It was able to outperform the other methods in terms of Fermi enrichment as quantified by the logarithmic AUC, logarithmic corrected AUC that you can see here, where the x-axis in the logarithmic scale to put more emphasis on the initial, the classification on the initial, the top of the rank that is established by the score you are testing, because this is more meaningful in virtual screening where you just test the top 100 molecules. Okay, so just to conclude a bit, I think like we obviously get a lot of information every day and a lot of milestone or like transformational changes about AI, but I believe that there is like an increasing potential for combining physics-based computational chemistry and AI in ligand discovery. It is not an aspect that we should totally neglect. The synergy is quite mutual because physics-based is generalizable, but is expensive to perform. It's expensive to run simulations. Even docking is very expensive. Machine learning, especially active learning or reinforcement learning methods, can accelerate the optimization of physics-based quantity, the optimization in the physics-based space by order of magnitude. And this is like very promising when we look for this kind of target that have no prior ligands that have been reported. At the same time, physics-based methods can generate large amounts of data for training. They can act as data augmentators, as for example, I hope I convince you that we did in DocBox, where fitting energies, binding energies, or binding affinities, solely in crystallographic structure will totally neglect the conformational landscape of binding events. And I do believe that harnessing the synergy is very crucial to advance the field, and this will be true until we have the comprehensive coverage of all the proteins that are druggable and all the chemical space, which is 10 to the power of 60 molecules. And therefore, I am quite sure I won't see that during my lifespan, but perhaps I will. But I think that for now, we're still far from having models that can generalize to the entirety of the chemical space without relying on a physics-based quantity. Okay, so with this, I would just like to acknowledge all the collaborators of the works I presented, various labs around the world, and obviously my lab members. Some of them are reported in that picture there. Like I said, most of the work done here is done by them. And of course, like the funding agency that have allowed me to perform these research and supported my scientific endeavor. And I also like to thank you for the time, for giving me this great opportunity. And thank you very much, the organizers. And if you have any question now, I'll be glad to take. Thank you very much.
Thank you for the presentation. So we can take questions now. So you can either write from the chat box or you can open your voice and physically ask your question.
So, sorry, Denise, should I just read what is
I can read,
in the
but
chat?
if
I
you
can,
can
okay.
read, you can choose both.
Okay, can we reliably use in silico data such as data produced from molecular docking experiment to train machine learning models. Generally, we prefer experimental data to build model due to its accuracy. How trustworthy are models built primarily on silico data? Are there any considerations when using such data of machine learning development? Okay, these are like very interesting question. One of the things I noticed while reading that, we prefer experimental data for building machine learning model due to its accuracy. I would first debate that experimental data is by no means accurate. In other words, there is a lot of variability when testing, for example, potency or like biological effect between an assay, even the same assay performed by two experiments. There were like a number of very interesting papers on the reproducibility of experimentally measured quantities. Therefore, in my opinion, for example, training a regressor based on for example, binding affinities coming from different assays, from different experiments, but all stored for the same target within Campbell, it's very challenging. It's very challenging because the noise in the experimental data. Now, training classifier is probably a bit easier because the order of magnitude should somehow be kept if we trust that. Like I said, I don't think molecular docking or any physics-based method can really replace that. Like can really replace like an experimental measure quantity. What I was proposing, what we were proposing, is to improve the features that you have. So improve, like do data augmentation in terms of like a feature that you train your model with. The representation you use to train your model can be, for example, a computational ensemble. And then to predict the binding affinities or binding affinity class based on that, because that will, in my opinion, is certainly more meaningful than train based on a smile or something that encode 3D 3D information in a very, very implicit way, very, very far, very, very non-obvious way. So that's my idea. But for example, like if you are interested in generative models, you will see that a lot of generative models actually rely on optimizing, on generate molecules and optimize them for docking scores. So there must be a reason why they follow that, is because especially if you look for new data, like I say, like in a new target, there is no way you can train a model for that. Or I mean, there are ways perhaps, but that optimizing for a physics-based quantity is probably coming from a computational method is probably the way to go. Okay, so the second one, thank you for the great talk. Thank you. I had a question about a deep dock to disorder protein. Since the structure are flexible and binding sites are well defined, how can deep docking be used effectively in such case? Yeah, this is a very good question also because they presented some interesting results, in my opinion, on amyloid beta 42 peptide, which is like intrinsic use as a protein. Now, if you, in that case, we're targeting like a putative site that is somehow ordered when they aggregate in fibrils in the secondary nucleation settings, so like the fibril is already formed, so the binding site is somehow the one we target somehow formed. There is no, like deep docking itself can be applied with any docking model, any docking program you want, and to how many pockets you want. So for a disordered protein, I'm not like an expert in that field, but I would assume that you can, for example, simulate the ensemble of the intrinsic allele of the disordered protein and identify suitable binding sites, and then you can, for example, train a model that can predict molecules that can bind multiple of the sites that are identified there. There was a paper I've seen recently published on this in like pointing at this direction. So if you're interested, just drop me an email. I can point at the paper. But I will say you can apply the same identical protocol. You just consider multiple sites. So then you will test molecules that are likely to score well in multiple sites. But again, deep docking just work as well. Like the maximum capacity has is like how good is the underlying model.
Yeah, are there any more questions?
Yeah. I'm wondering about when we have docking, let's start with the docking or other batch docking with code. Should we recalculate the binding edge score and MSD value with another in silico tools to confirm the results? So, okay, I'm not sure I understood that well. So the docking output a class, so it will tell you basically if your molecule stands, for example, in the top 100,000 molecules, top rank 100,000 molecules within your library without seeing a library. If you mean like, but what I understand here is basically if you have like just one docking tool, if you should interrogate your molecules that are likely to be good with one program, if it's worth to dock them with other program and then calculate the RMSD, like compare the two scores, so consensus scoring, and compare the RMSD. If the RMSD between the two poses is below 2, then you assume that is like a more reliable. This like goes under the name of consensus docking. And yes, like a couple of the cases I showed, like we're using that, and in all cases we always have used like a multiple scoring function. So MC we use Autodoc and MGVSA. I mean, beta was a Fred and Lina. And then like in the other I presented with like actually consensus document where we also like compare the RMSD between different poses. So yeah, I mean, it's a hard question because sometimes it works very well. Sometimes no, it depends. I will say like it depends if you are, it goes a bit a luck. Like if like consensus capture the artifact, like the holes in the scoring function of both, and then you're just getting with molecules that are just badly predicted consistently by two methods, or if you're actually able to capture intersection of where the two scoring function and two docking methods do well. So it's a bit hard to know.
Thank you so much. I'm sorry. I also have similar along these lines a question. So if it is okay, can I go and ask?
Yeah, for me, yes, for sure.
Thank you so much. First of all, it was fantastic. And so there are two types of consensus, like, right? One of them is consensus scoring. One of them is more orientation. You are checking the orientation with RMSD, and it is consensus docking. So when you apply, I think I see papers debating like which one is better, and some of them are using both of them together. But when you have both of them together, sometimes you are ending with nothing. So in your experience, which one do you think is seems to eliminate the artifacts better without before going into the MD kind of validation?
Personally, like if I have like empirically based on what I've seen, I would tend to say that consensus scoring is probably like safer.
Okay.
But there, with a caveat, the problem with consensus scoring sometimes, and this is something we have seen, for example, in the development of DocBox 2, where we generate conformational ensembles, but we use multiple programs, and then we do consensus scoring. So, for example, we have pose from Bina, pose from Doc, pose from Boltz, pose from AlphaFold. But then we rescore each pose. The features are actually the score, for example, the rescoring of all the poses with each scoring function. So the pose from Bina will be scored without the scoring function, the pose from Boltz will be scored without the scoring function, and so on. The problem is like some of the programs, they tend to make poses that are favorable for the program, but very unfavorable for another program, because of clashes. So there are clashes, like numerical errors and like clashes that are seen by a program, not by another. So you can end up with like a pose that is perfectly fine, even for an active crystallographic structure, that is barely scored by one of them and well scored by another, well scored by the one that generated the pose and badly scored by another one, even if it's like a pose below 2 Armstrong from the crystallographic one. You don't know that, but like if you do the retrospective experiments, you will know. So what we resort to do before doing consensus scoring was to actually minimize with a unbiased scoring function. So we use, like, for example, Amber, like the Amber molecular dynamics engine, to just like do a relaxation of all the poses, constrained relaxation, but then just to remove these clashes, because molecular dynamics force field are better at detecting clashes. So then you have like a more fair rescoring. Because otherwise you end up with poses maybe are good, but are just like scored plus 100 by one of the programs just because it sees some clash and he has some numerical error that the other doesn't see. So like the only problem with consensus scoring is that you always need to kind of somehow minimize the pose a bit after. But I will say it's probably because you rely less on the sampling of both algorithms and like what both algorithms come up with in terms of sampling. Because like I said, there is a problem between sampling and sampling and scoring. In 2019, we did a study. Like I was still like a student, but we did a study on how to actually do consensus talking, but by using rescoring in a way that you kind of like overcome the limitation with pose scoring after sampling. And if you're interested, I can send you that paper. We had the code and everything. This DocBox 2 is an evolution of that. Is an evolution of that scoring, which basically you don't need to worry anymore about you don't need to worry anymore about the sampling of each program. Everything is considered the same time. So that's my idea. As in terms of RMSD, sometimes works, sometimes doesn't.
Yes, I agree. One like maybe a continuation. So when we are talking about scoring, because different programs have different energy functions, right? And then they give like quantitatively they give different scores. So when we are talking about consensus scoring, we are talking about actually the like when we sort it where it is, right? Not like
Yeah.
Okay. Yes. Okay.
Well, yeah, it depends. So I think I presented one of them where we're doing voting. So that's exactly you rank and then you see where it is. The other way you can, like the other way to get done is like a normalized or standardized within each score. So you standardize each score and then you can average on that because at that point they're all like,
yes,
or normalized, even better. Maybe if you have similar distribution, like normalized, so you adjust that between zero,
yeah, I
you
see.
have just the same range.
I see.
0, 1, and you can but where they are ranked on each score is like the most straightforward way to do that.
I see. And I see iterative process, you are using different scoring functions, right? And
yeah,
for example, in the emulate beta one, you were only using one of them to train all your model to like this iterative process is going. I know you have different models. At the end, you are eliminating some data set and then you are filtering them out. Which one do you think is better? And then also, like in similar lines, I have this question. Do you think that it is better to filter out the chemical library before starting the deep docking or after starting the finishing the deep docking? Like with respect, like I'm thinking of more in lines to not making model like to add some kind of perturbation to the model to not make it learn something that may already be a hit. I don't know if you
Sure. By the way, there is someone on the queue, so we might like.
I can stay longer for the questions so like I can answer. But if you want, yeah, I can answer another question before I can whatever you prefer, Busra,
So
I can
like
answer
if we can continue a bit later with Bushra's questions, but asking so dear Havan Hocam, you can ask your question.
Hello, thank you for your presentation. Actually, my question will be the cache challenge. I am interested in the Parkinson disease, and especially for your project, it's lesson reach repeated region 2. You have a project about the cache challenge. And I'm curious about what was your starting point about in this challenge? Did you know that any known inhibitor or any ligand, or you first did just structure-based drug screening or something like that?
Yeah, okay, so that's a good question. So in the challenge, basically we don't have any choice about the target. They propose the targets, and those are selected by an external committee. So I'm not an expert at target, but the LRRK2, there are like molecules that inhibit the kinase domain. But those, like most of them, stabilize the open conformation, if I remember well. So like the idea here was actually to target the VDR domain. Was inhibition was like a function is not, at least at the time of the challenge, I don't know how it is now, but it was not well known, but likely promoting the binding with microtubules, if I remember well. Therefore, for this domain, there were absolutely no known ligands at the time. So it was a purely structure based, purely relying on docking. So like whatever bind well in the central cavity. I think at the beginning of the challenge of something we look in was like there were other VDR domain in the PDB, and there were other ligands that were reported for so similar folds. And we tried to see if we could like train some models, but like I said, we weren't really confident into that because it was also like for different target. The only thing being like there were similar fold of the Woody 40 repeat domain. So we resort to just like optimize for docking scores. And yeah, our starting point was the crystal structure of Woody 40 domain, and that's it. And we look for things that will we look for molecules that will bind computationally tied to that.
As I understood, you start with the similar structure actually because you don't have any ligand at the beginning, and you look the other proteins which have VDR domains, and you look the other ligands which are binding the other proteins, and you trained your machine learning with this data set, and you predicted for your protein.
No. We wanted to do that. We looked into that. We looked into other like different VDR domains, but then we end up doing nothing with that. So we didn't use any of this data. We didn't try because it was also like a very small amount of data and very sparse between proteins, different proteins. So it would be harder to train something like even selective for the VD 40. So we end up just relying on docking.
Thank you.
Okay.
So if you like, you can.
Yes. So I also don't want to take a lot of your time, but okay, maybe this would be easier. Do you remember my question, or should I again ask it?
Yeah, I remember.
Okay, then can you?
So I remember like so the first one you ask is like when we screen a library, then we basically predict multiple scores, and then in some of them we just predict one score. So this was more like practical for cache challenge because we start with a very small library. So the idea was to do what we earlier have done for SARS-CoV-2 main protease, which was basically try to do deep docking with consensus scoring. So you start a library, and your library is pretty big. This one was like 4 billion molecules. And we use Autodock GPU, which is the fastest of the docking programs, to try to reduce the library initially. And then we end up with a pool of molecules that are predicted to be top scoring, but there were still like a significant number of molecules. From this pool of molecules that score well by the machine learning model, predicted by the machine learning model, they were scoring well with Autodock. Then we use that as our initial library for the second method, and we did it with the second docking method. So we keep doing that in this way. Every time is reduced, and then you try to find the intersection. It was just to reduce to a lot of molecules from a very large library because 4 billion is very computationally demanding, and then end up with a small amount by leveraging these jumps where like you predict all the top scoring for Autodock, and then you jump into, and then you have this one, and it's much smaller library to work with. And then those molecules also, of course, they are supposed to score well, but again, since they're all predicted scores, you don't have any certainty about if the they are scoring the same pose or similar poses. So I would say it's more practical when you have this large, and probably there are better ways to do that. I mean, definitely you can improve the deep docking model itself to deal with much larger libraries. In terms of, like I said, regardless of like if we predict or not, in all the cases we always have used RS scoring method, because even in amyloid beta 42, the poses got from Bina, then we, or the promising molecules, then we check the Fred score, so the Ken-Gauss scoring function from Fred docking, and use that to basically rescore the poses and select the one that score well in both methods. So for the very large library, we just did that sequentially with prediction because we couldn't dock that many molecules. But in general, like I say, I think it's good to keep like a multiple scoring, consensus scoring method.
I see.
And
Okay.
I don't remember the second question because I think there were two.
Okay, yes. The second one is this. Is it better to filter out because we are all druggable sites and stuff.
Yes.
Filter out before or later?
I think it's better before I mean, in general, is always better before. The thing is, like, especially deep docking, the way it works with active learning is basically using an activation function where the molecules that are likely to, they have a high probability of scoring well, they're then taken and put back in a training set. If you have an artifact that you don't want but scores very well, this will be like amplified. For example, something you don't want from the chemical point of view. I'm thinking, for example, if you're looking, like, for example, like I show in the challenge, one of the goal was like to identify molecules that were not charged, right? So we didn't exclude those necessarily because we just want to see the entire whatever was coming out from the screen. But if you're doing like a project where you want to avoid having molecules that are charged, for example, you can compute the protonation states before and remove those right away or predict the charge. So yeah, and the other thing is chiral centers. This is practical because from these ultra large libraries, if you have molecules that are highly chiral, then when you test the molecules, you are going to test arsenic mixtures, and therefore, if you have molecules that have 4 chiral centers, then you have 16 isomers to test, and your potency may be diluted a lot. So you will rather like personally, I usually would just like exclude those. But I would definitely say if there is anything just based on the molecule that you don't want, like some molecular property of the molecule that you don't want, like charge, chiral centers, log P. If you know that your molecule that your target doesn't, you need specific properties of the heat, then it's definitely better to do that before. Okay. Because then you won't learn that part, which is like, it's not something you want to learn from.
Okay, I see. Thank you so much. Thanks a lot. This was really fantastic. Thank you so much.
Thank to you. It was very nice. I don't think I have any other questions.
Yes, I also couldn't see any more questions. So if it's feasible, I can end this session. So thank you for coming and presenting your topic to us. And
thank you.
We really thanks so much to you. It was very good, and I feel honored. And if anybody has any question, like feel free to email me. I try to answer as fast as possible. Okay.
Thanks a lot.
Thank you so much.
Thank you. See you hopefully.
Bye bye.
Bye bye.
See you soon. Hopefully see you soon. Bye bye.
Bye bye.